Biotechnology has opened doors for many applications in the fields of agriculture, therapeutics, diagnostics, waste treatment, food processing, bioremediation, and energy production. It uses genetically modified plants, fungi, and animals to produce biopharmaceuticals. Biotechnology operates in three critical areas of research. First, it provides the best catalyst in the form of improved organisms such as microbes and pure enzymes. Second, it creates optimal conditions for these catalysts to act in. And third, biotechnology provides downstream processing technologies which help in the purification of protein and organic compounds. Biotechnology also plays a key role in agriculture and food production. With a gradual increase in the world's population, there is a growing concern about increasing the production and quality of food. Traditionally, agricultural production can be increased by employing two methods: the use of agrochemicals and organic farming. During the Green Revolution in India, crop yield had increased substantially due to better management practices, improved crops, and above all, the use of agrochemicals such as pesticides and fertilizers. The Green Revolution managed to triple food supply. However, it still fell short for the growing population. The fact remains that in developing nations like India, agrochemicals are still an expensive proposition for farmers. Besides, it is also not possible to increase the yield of existing varieties using methods like conventional breeding and organic farming. In such a situation, genetically engineered crop-based agriculture is a possible solution. Genetic engineering techniques have been used to alter the genes of many plants, bacteria, fungi, and animals, which are then called genetically modified organisms or GMOs. Nowadays, genetically modified or GM plants are being used increasingly as they have several benefits. These plants have a higher tolerance to abiotic stresses such as cold, drought, salt, and heat. And they also help reduce the use of chemical pesticides, as many of them are pest resistant. In addition, GM plants increase the efficiency of mineral usage and thereby prevent early exhaustion of soil fertility. Moreover, GM crops, such as vitamin A-enriched rice, enhance the nutritional value of food. Some GM plants also cater to industrial needs by providing resources such as starches, fuels, and pharmaceuticals. However, the greatest contribution of biotechnology to the field of agriculture is the production of pest-resistant plants. For example, Bt toxin produced by a bacterium called Bacillus thuringiensis has been cloned and expressed in many plants to make them insect-resistant. Bacillus thuringiensis produces some crystal proteins during a particular phase of its growth. These proteins, called Bt toxin or insecticidal proteins, are toxic and inactive. Although the Bt toxin protein exists as an inactive protoxin, once an insect ingests it, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilizes the crystals. The activated toxin binds to the surface of the midgut epithelial cells of the insect and creates pores, which cause cell swelling and lysis and eventually kill the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into many crop plants such as cotton. This variety of cotton is also known as Bt cotton. The choice of gene depends on which pest is targeted. For example, a Bt toxin is coded by a gene called Cry. Cry genes are encoded by Cry1AC, Cry2AB, Cry3AB, and Cry3BB. They provide resistance to cotton bollworms, corn borer, Colorado potato beetle, and corm rootworm respectively. Over the years, biotechnology has given rise to several interesting methods of pest resistance, such as RNA interference. It is a method of cellular defense in all eukaryotic organisms. It is designed to control the infestation of nematodes, or roundworms, which act as parasites in several plants and animals. For example, a nematode named Meloidigine incognitia infects the roots of tobacco plants and leads to a drastic reduction in crop yield. This pest in tobacco roots can be controlled using RNA interference. This method involves silencing of a specific messenger RNA of the nematode due to a complementary double-stranded RNA molecule. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements called transposons. These transposons replicate via an RNA intermediate called reverse transcriptase. Using agrobacterium vectors, nematode-specific genes are introduced into the host tobacco plant. Since these two RNAs are complementary to each other, they form a double-stranded RNA that initiates RNA interference. The introduction of DNA is such that it produces both sense and antisense RNA in the host cells. The small interfering RNA strands of double-stranded RNA bind to messenger RNA and prevent translation of the mRNA that cause silencing. As a result, the messenger RNA of the nematode is silenced. Consequently, the parasite is not able to survive in a transgenic or genetically engineered host expressing specific interfering RNA. Therefore, the transgenic plant gets protected from the parasite. Pests are the bane of food-producing plants. The growth of pest-resistant plants and the development of methods such as RNA interference have revolutionized agriculture and ensured good food production. The recombinant DNA technology has played a crucial role in the field of medical science and healthcare by helping in early diagnosis and treatment of many diseases. It has also enabled mass production of several effective therapeutic drugs. However, the most striking feature of recombinant therapeutics is that They do not provoke the human body to produce antibodies. On the contrary, use of products isolated from non-human resources can induce an immunological response from the human body. Over the years, biotechnological processes have led to the development of revolutionary medical methods and techniques. These are genetically engineered insulin, gene therapy, and molecular diagnosis. One of the biggest breakthroughs in medical science came with the production of genetically engineered insulin. Insulin is a hormone which regulates the glucose and energy metabolism in the human body. Insulin produced by diabetic patients is either insufficient or impaired. Therefore, they are given doses of insulin at regular time intervals. Earlier, insulin was extracted from the pancreas of slaughtered animals such as pigs and cattle.